Hello, today we're going to tackle the DNA replication notes. I'm breaking the video up into several videos so that it's more manageable and you can go at your own pace. But you should have your note template that has kind of fill in the blank stuff. So if you wanted to pause the video first and fill in what I have here typed in red and then resume the video so that you can then listen to me, that might be a good idea. So go ahead and pause the video if you would like. Okay, so the summary steps that you've just written down basically summarize what we're going to be getting into in detail. So it says the two DNA strands of the double helix unwind and separate, right? We say that the parental strand, so like the original strand, is used as a template to form a complementary. There's two parental strands, right? The strands are originally attached by hydrogen bonds in between them, so we need to separate or break those hydrogen bonds. And then the parental strand and then the new daughter strands wind together. And so when we did science practice with replication, we found that this was called semi-conservative replication. So again, this is just an overview. We're going to get into the details. So rules to consider. Again, if you would like to pause the video so that you can fill in your notes and then resume when you're done writing this red portion down, then I'll talk. Okay, so rules to consider. To achieve polymerization, right? So we're making a polymer, we're making DNA. DNA must occur in a fixed order. Sorry, DNA replication must occur in a fixed order. DNA polymerase, so here's an enzyme, ASE, enzyme, can only add nucleotides to the 3' end of a previous nucleotide. So we have to remember about what we learned about the structure of DNA with those notes. DNA is anti-parallel. That means that one strand, okay, so if it's double-stranded, is 5' and then the other side is 3'. The other strand of DNA runs in the opposite direction. So when we did the history notes, we said that the phosphate group was attached to the 5' end, but the 3' end did not have any phosphate attached. So if we have phosphates on this side, it makes sense that you can only add new nucleotides to a free 3' end. So stick them on here, stick them on here, stick them on here. So we say that the new strand is synthesized in the 5 to 3 direction, right? It's elongating in this direction. So if I were to add nucleotides to this one on the bottom, they would be added here and here and here and here. So synthesis happens in the opposite direction. Okay. The template strand is red in the 3 to 5 direction, okay, because you have to look at what comes next. So 3, D, okay, so 3 to 5, okay, but it's synthesized in the 5 to 3, so it's elongating in this direction. Okay. DNA polymerase requires an RNA primer to begin building a new strand. So what we need to look at is the structure of a nucleotide, and then we're going to revisit what does this mean to be an RNA primer. Okay. Let's label a couple of things on this nucleotide. Right? So first of all, you should recognize that this is a picture of a nucleotide, and it's got 3 components, right? Sugar, phosphate, and base. A sugar, a phosphate group, and a nitrogenous base. So in this picture, this arbitrary box represents a base. Okay. This pentose structure here is your sugar, and then P is your phosphate group. Now, when we're talking about the sugar, we have to identify that the carbons, okay, 1, 2, 3, 4, The fifth one is kind of tricky, ends up being out here. This could be an oxygen. Okay. So we label these carbons in order from 1, 2, 3, 4, and 5. So the base is always attached to the first carbon, and the phosphate is always attached to the 5. That's why, again, when I drew this circle here, this is that phosphate attached to the 5' end. That's the fifth carbon on that sugar. Down here is referring to this available sugar here. So new nucleotides can be attached here. Okay, there's usually an OH group right there. On this 2' hanging off of here, it could be H or OH. If it's hydrogen, that means it's missing an oxygen. So this is the deoxyribose. This is the sugar in DNA. That's how it's different. If it's OH, it has the oxygen. So then this is ribose sugar, and that's going to be found in RNA. Because both of these are nucleotides, because both of these are nucleic acids, they're both made out of nucleotides. Okay. Bases, right? Let me start over. In a base, for the bases, your options basically include A, T, C, and G if we're talking about DNA, or A, U, C, and G if you're talking about RNA. Thymine is replaced with uracil when you're referring to RNA. Okay, so again, this is a whole picture of a nucleotide. We're looking at the parts, identifying what are the carbons, and this is where a new nucleotide would attach to a free 3' end. So again, when we're labeling DNA with the ends, this is what we're referring to. This is 3', this is 5'. We're talking about the parts of the nucleotide. Okay, I'm going to stop the video here for right now, and then you can go on to the next if you're ready.